We'll start. ADR, all these recordings. So it's our actually last in-person colloquium of this semester, and it is planned to be this way. Next week, pay attention that it is the colloquium is part of the Israeli physics colloquia series. So it's going to be online, and there's a great speaker. I don't remember who. Penrose?
Not greater than this one.
Penrose? Penrose, yeah. But today we also have a great speaker, so it's my pleasure to introduce Ro'i from Tel Aviv University. And first, thanks for those who still are here in person. Great that you are still coming, and thank you, Ro'i, for being here in person. So he's a biophysics, soft matter physicist from experimentalist from Tel Aviv University. He started his way as in Deutsch's lab as a condensed matter physicist, and then found the light or the life and
softened.
And did his postdoc in Santa Barbara. And from 2010, he's back in Tel Aviv University, studying various aspects of genes and other words.
That will be explained.
That will be explained. And you'll talk today about how you find order from disorder. So thank you for taking yourself.
So thank you, Amos, for the introduction, and it's a really great pleasure. Just reminded today that my first paper ever in the academy was when Amos was the first author, when he was in Jerusalem as a graduate student, and I was still an undergrad or something like that. So it's a real thanks for the invitations, and I'm excited to be here and tell you a little bit about why I find biophysics, in a sense, interesting, what's the physics in it, and some of the stuff that we found, some of the stuff that others found. So I'll try to make it into a colloquium-based and not a seminar specifically in biophysics to show you the light that I at least found in this in some particular topics there. So let's go on. So I'll start very broad. So I know that since Amos invited me, so I know that it should be broad enough for all physicists to know a little bit about the language that we are using in biology. So the central dogma, in biology, they have really big names like dogma and paradigms and all of that. So the central dogma is that we have our, why is that not working now? So we have our DNA, here it is. Okay, so we have our DNA, I need to point here. Our DNA that is being transcribed to another molecule, which is called an RNA. These are four-letter linear code. Right? So there are 4 different types of molecules that are stretched one after the other. These are our genomic code that encodes all the instructions to life. And those RNAs are being, after that, translated into a protein. A protein, it's made out of 20 different types of molecules called amino acids. And they make up, again, a linear code, one after the other, according to the descriptions of those 4 letters in the beginning. So 3 nucleotides coming from the RNA are being translated to a specific amino acid, and then we have another linear code. So what do we do with this linear code? So this linear code is made out of this amino acid that I told you before. You see there are many types or many different structures for those molecules of the amino acid. Some of them are charged, some of them are hydrophobic, some of them are small, some of them are large. And they make up into like a linear polymer. This linear polymer can be very long, can be the average size of a protein made out of this would be about 500 amino acid. So the combinatorial options that you have to make a protein is very large, right? Much larger than one. And but something very surprisingly happened. Although that you have all of those possible combinations to make up a protein, apparently the central dogma at least, tells you that they will fold into specific structures, alpha helices, beta sheets, these are the names that we have. And those secondary structure will further on make into a structure. And this structure, this three-dimensional structure is unique. That's what the central dogma tells us. And it actually tells us the code to make up a protein. Yes?
Is it the same number proteins in all different pictures, but if you take terms or basic pictures?
So that's an excellent question. So the answer for that is yes and no. So that's a good answer. So you will find that specific proteins will make up a large chunk of the proteins will be conserved throughout evolution. And then there are parts that will be specific to the specific organism. So you will find some basic units in all of the creatures that are identical, and then there are some diversities.
And will the identical ones be simpler than the diversities?
Yes. So you will find some basic units in all living creatures for the basic functionalities of our cells, since the cells of a microbe is not very different than our cells. I mean, it is, but not very in terms of numbers. Okay, if you would count how many different microbes. So we have gazillions of different types of protein in our body, and there are some that are very, very conserved throughout evolution. And this is, by the way, the way that we can know that evolution was actually there, right? Because we can see these motifs in earlier and earlier creatures in terms of the evolution tree. So why is that the structure is so important? And that dates up to quite long time ago to Emil Fischer, one of the Nobel laureates, a famous Nobel laureate in biochemistry. And he coined this term that called the lock and key model back in 1894, really long time ago, more than 125 years ago. And the idea is that you have a structure of a protein. Let's call this protein, specific protein, let's call it an enzyme that has some functionality. And it's a lock that will find another protein. Let's call it a substrate. It's just names, okay? And then they will find each other according to the structure and will fit like a key finds a lock. And once they are connected, once they are interacting one with another, something happens, right? I mean, the lock can be open. Some structure is being made. Some chemical reaction is being constructed. And that is one stroke in the domino, meaning this result in a sense to a functionality. A functionality meaning that something happened and now another thing can happen, and it's domino of actions because of recognition between one protein to another, makes functionality in our cells, yada, 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 we have Amos, okay? Meaning that all of these functions, one after the other, will lead up to all the functionality that we have in our cells. So that's doing like Bio 101 in 5 minutes. And this also explained to you why mutation, changing in the amino acids, makes in one case disease because the key now doesn't fit into the lock, or can make diversity. Now we are, we have a better fit to meet the environment, so we can evolve. And this is maybe answer also your question before. So you can have some small mutations in order to accelerate your fitness to the environment. And this is why creatures become more and more evolved over the evolution. Okay, so that was the basic thing. Now, how can we, so that immediately tells us that the structure has a lot to do with functionality in biology. So a lot of people try to find out this singular structure We start with a very long polymer, but somehow it finds its way to a singular structure. So there were a lot of techniques that were developed in order to find this structure, including crystallography, NMR, and very recently, the major hit is doing electron tomography in order to find what is a specific structure of a protein. This is the protein that Ada Yonat got her Nobel Prize for. It's a part of the ribosome. It's a protein that makes protein. Okay, it's the machinery that takes RNA and amino acids from a solution and make out of them these linear chains of other proteins. So it's a very, very important protein in our body. And this is another protein that we all heard about recently. This is the spike variants of COVID. Okay, and you can see here alpha, beta, delta, and kappa. Kappa didn't make it into the news, apparently, but Omicron did, and I didn't find its structure yet. But you can see that there is a minute mutations, changes in the code of these proteins that makes it to be more infectious or more deadly. Okay, so this is something to remember when we are talking in the news and we are hearing all of these talks about mutations, what they are doing. This is what they are doing. They are changing a little bit the structure. So one of the biggest advances, that were in the last couple of years, in my view, in biology, was actually this revolution. So for a long time ago, we were thinking, a lot of physicists thought, okay, you have an amino acid, give me the sequence, I will tell you what is the structure, right? I mean, we have very smart people like Yitzhak here. They can do a computer simulation and tell us that they fall to like that. And if they fall to like that, okay, now I know how to treat that with a different protein that does like that. And then we can solve all diseases in our body. It's very important. What is antibiotic? Antibiotic is a small molecule that get into these ribosomes of Ada and disturbed it. And but it disturbs only those that are ribosomes of bacteria. So this is how it works. So if we know the structure, we know how to fit things into it. Apparently, this is a very, very complicated problem because these molecules are super complex and they are in water and they are in room temperature and everything is moving. And this problem was so hard that nobody could actually solve it properly. Although they are folded into like secondary structure and the secondary structures fold into more, it was an unbelievable difficult problem. And this difficult problem was apparently solved just a year ago, 2 years ago, by this group of people from DeepMind. DeepMind is a subsidiary company in England of, this is not me, right? Okay, of Google. And they develop a machine learning algorithm that you can plug into it the amino acid sequence, and it folds. It folds. This molecule will tell you what are the positions of all the atoms that you have over there in order to resolve the structure. So it's a computer generated with no simulation, just through training of a machine learning algorithm. And this is quite amazing. Today you can open up, they made it publicly available over the internet, free of charge. You can just plug in the sequence and it will give you what is the structure. So this is one of my favorite proteins. I'm joking, I don't have any favorite protein. This is hemoglobin, subunit theta 1, just one that I just plugged in. And you can look into the protein database, and there is also a link over there that immediately gives you the how does this protein looks like. You see, this is how it falls into a secondary, ternary structure of these alpha helices. And what is more amazing is what you see here on the left. What you see here on the left is actually what is the confidence that they have in this structure. And you see that everything is with a confidence of a few angstroms. Yes, Eli?
This is theoretical or experimental?
This is theoretical, and it matches, it was trained through experimental, and the level of confidence that they have is coming from how much it is matched to experimental And they know how to do it now for a lot of proteins, so they have this confidence level checked out. And this is available through the internet today. You just plug in sequences and it gives to you immediately. Yes, Elie?
So is the correct folding a global minimum, or is it a local minimum? Can you now, when you know how it folds, can you go backwards and ask this question?
It's a black box.
This is a black box. This is a
no, but you
machine.
know how it folds. So
No, you don't know. This is, you put a sequence and it gives you the structure. It gives you the three-dimensional structure. What is the position between each amino acid in 3 dimensions? This is not a simulation. What we are thinking that in many, many cases in biology, the structure, the 3-dimensional structure of a protein, it's a very, very deep minimum of the global minimum. And there are some cases when you have local minima in the energy landscape, like an intermediate. But once the reason that we are more confident in that is that proteins, when it's in its structure, it stays there for a very, very long time. And it's very unique, meaning that if you will throw up a lot of them, all of them will fall into the same thing and will not fluctuate between, other minima. Energy, energy, okay, free energy. So if you think about the coordinate, the coordinate systems of all the positions of the molecule, you will find one with the lowest free energy to it. So that's why it will be in this structure.
But was the experimental data used in the training?
Sure.
On this protein. So, but what about predictions?
No, so they have predictions for all the proteins of human protein. All of them were already predicted by that machine learning. So they took okay, so what DeepMind did is took the entire database that was experimentally verified of tens of thousands of proteins that we already know by very stubborn and talented biochemists like Ada, that resolved the structure experimentally. And they took these data sets and through a very ingenious path, by the way, they made a machine learning that can take you not just those that we know experimentally what is their structure, but also those that we don't have a clue about what is their structure, and they can now give you the 3-dimensional structure.
So was it ever checked?
Yes, obviously no. So what you do is that you take a part of those proteins that you know their structure, and you train them over there. And then you put another set that was not part of the training, and you verify that this is actually give you the same prediction. So you never take the entire package. Okay. Yes.
Kind of related to that. So you said that there are of the order of 500 to the power 20 different possible proteins, basically an infinite number. How many proteins do we know that exist in, let's say, the human body?
Some very, very limited, small number of those combinations, about a few 100,000.
Yeah, so you're talking about
a small fragment. Yeah, a very small fragment. Yeah.
Okay. Okay.
And the question why? The question is because many of them will not be functional biologically. So there is no reason to keep them up. Okay, that's the logic. Okay, so only a fraction of them can make it into something useful. And biology or evolution usually throw away those that are not functional. So if it will make me a horn and I can use a horn, I will have a horn in the end. So that's the logic. You had a okay. So again, as I said, this is not my favorite protein, although it's a very nice name in the database. So what is my favorite protein? This is actually one of my my favorite protein. It's called the neurofilament light polypeptide. It's a protein that I was studying in the last 15 years, so this is why I like it. And what you can see already here is that a lot of these regions has a lot of confidence, very high confidence of the model, but there is a lot of yellow and even red positions here. So this is a failure of this magnificent thing that was just developed like a year ago. Meaning that in their scored residue, they are already telling you, listen, this one, we don't really know its structure. And this is a problem, right? Because I just told you that we solved the problems of 50 years, and now I'm telling you that we didn't. And in fact, this is true. In fact, in the last 20 years or so, we realized that this paradigm that we have into sequence, structure, function is only partially true. Meaning that there is a lot of proteins in our body that have a sequence of amino acid, okay? But they do not fall into secondary structure, okay? They do not fall into secondary structure. They do not fall into ternary structure or quaternary structure. They remain as polymers. The term for them is intrinsically disordered proteins, meaning that they do not fall into something. They're still disordered in solution. And what is more surprising is that about half of our protein have large domains that do not fall into secondary structure, meaning that they are disordered. They are fluctuating between different minima in the energy landscape between one conformation to another conformation. Half of our proteins. We had dogma, we had paradigm, we had all these beautiful words. It fits very well to half of the problem. So in many years, this is something that I was studying and how and why we need these intrinsically disordered proteins in our body. And the reason for that are multiple functionality that we can achieve from disorder. For example, I told you that we have like a lock and key mechanism. But if you go to your janitor, he has a master key, right? That can open up all the doors in your corridor. This master key, you might need to jiggle it around a little bit, but it can open up a lot of locks. Meaning that those proteins are known, are notorious to interact with many different partners, with very weak energetics, but to interact with many of them. They know how to make through this interaction to make beautiful things like liquid-liquid phase separations or aggregations or some kind of passivation or filtration, just being a polymer that can move between a lot of different conformations. So one of the biggest hits that we have in the recent years is this mishegas about liquid-liquid phase separation. Biologists found out what I think physicists discovered like 60 years ago, 78 years ago about phase separations of liquids. We know it from vinegar and oil, you know, that they are phase separated, still becoming liquids, right? It's not something very dramatic, but apparently it's a big hit in biology because you can make by this kind of very weak interactions between disordered proteins and RNAs, you can make membraneless organelles, meaning machineries that are phase separated without the enclosure of a border made out of membranes. And this makes a lot of impact into biology, finding out how you can use those or how nature uses this kind of membraneless organelles for all kind of functionality never thought of before. So we have more organelles in our body than we thought of. Where we contributed some was to the understanding about those disordered proteins when they are packed into the cytoskeleton, into the skeleton inside the nerve cells. So here that there are these proteins that I told you. Remember neurofilament flow that I told you I like it very much. It's a long protein. Okay, all of those proteins that have very, very extended disordered domain. And those disordered domains are making the entire self-assembled structures to be something like a bottle brush, something like a cylinder of about 10 nanometer in diameter with very long arms. And those arms knows how to interact. And you can see here like a cross section from the nerve cells that are making this mesh-like, this jello-like structure that are very important for our nerve cell in terms of mechanical stabilization. And again, as I said, there are several proteins. They have a very long tail that is disordered, and it make up this the self-assembly to the ordered structures to make this bottle brush-like filament. Now, and what is very interesting is that although that you have disordered domain, something that doesn't fall into a very rigid body structure, it makes on a macroscopic scale a very well-aligned and regularly spaced liquid crystalline structure. And all the interaction is mediated through this disordered domain, this disordered tail domain. And this is something that got us interested. Yes?
Just to understand a bit better, is distorted domain, is it flexibility of this molecule? So let's say my arm, I want to twist it. So I have some part of it which I can twist here and here, so it's a bit more flexible. And similarly, you have some large molecule, and part of it is more flexible, and other part is less flexible.
Okay.
Is this more than this?
Much more than that. So if we take a structured protein, the level of the mobility, how much the molecules move, is in a angstrom level. So how much, and that's the resolution that you have in crystallography. You take those proteins, you crystallize them, you evaporate all of the water out of them, and you have a resolution of where is each atom in this ginormous, million of atoms molecules up to an angstrom resolution. So we know each one of them is there, and it's moving by no more of 1 angstrom. Here, the fluctuations of structures are on the nanometer length scales. So orders of magnitude more flexible. They can be, and they can find they're not standing still. Now, the protein, what is very surprising, the proteins, those structured proteins, as crystals and in solution, they have the same structure, meaning that they do not change their position because of flexibility or because of the environment, hardly any. So, the level of flexibility that you have in your joints is coming not because of your bones. It's coming because of your ligands and because of the water inside it. Okay, that's what gives you the flexibility. It's like saying that your bone will be able to do the same flexibility as what I'm talking about here. That explained to you? Okay. So let's continue. So what was the question here that got us interested? So one of the questions that got us interested is that we take all of those protein that I told you, we self-assemble them into these butter brushes, okay, these butter brushes that we have over here. And we want to ask what happens when we induce stress into them, when we try to compress them with osmotic pressure. Okay, how much they are stable to maintain the structure. And here you can see experimental data of, I didn't give you the legend here, because there are gazillions of different conditions, meaning what are the compositions of the protein that you have in the system, and what is the environment, what is the salinity of the buffer, and many, many different conditions that we tried over here. And we put them all on one master curve. And what you can see, the x-axis here is what is the distance between 2 filaments that we measure through X-ray, and what is the osmotic pressure that we induce on them. And you can see, so this is like when we teach in undergrad, what is the pressure volume diagram, right? I mean, this is the canonical statistical mechanics diagram that we do. And what you can see is that we have 2 regimes. We have one regime when it's in high pressure, we kind of see a universal trend. Okay, everything doesn't matter what are the details of the experiment, and I didn't give it to you. There is a universal trend in the pressure versus volume or distance in this case. However, at low pressure, we find 2 different trends. We find one which is more expanded. Okay, the distance between 2 filaments is more extended, and one when it's a little more condensed, right? I mean, the distance is more close by. And we try to understand it from so we did the measurements, but we also wanted to understand the physics behind it, because those are polymers. And polymers we should have known for many, many years. This is something that statistical mechanicists studied from the 60s when Itzhak was young. Okay, so so this is a problem that we might be able to resolve. So we start with a high pressure when we see universality, and when you see something that is universal, you immediately think, okay, this is where mean field theories should be dominant, right? Because the details of the experiment doesn't really matter. So how do we do that? We take these butter brushes and we put them in a hexagonal lattice just because it's easier, not because it's really they form an hexagonal lattice. They are more in a nematic liquid crystals, but let's do this approximation. And then we forget about the backbones themselves and we just think about the polymers in between that mediate the interactions. So we treat them like a semi-dilute solution of charged polymers because these are extremely charged polymers. And so from charged polymers, we know what is the pressure, the osmotic pressure that they are inducing just because they are charged. And this is something that we know. And we can also treat the fact that they are occupying space. So we can treat them a little bit more, do some kind of a Mayer expansion and do like steric repulsion. So all the things that I gave you now are some repulsion resulted from these charged polymers in between. So this is to a first approximation. The thing is that once I'm doing that, I know everything. I don't have any fitting parameters because I know exactly what is the charge on each of my molecules. I know exactly what is the length of those polymers. I know exactly the compositions in each of my experimental conditions that I measure. I know what is the salt condition, so I know how I spin them up. I know everything. So let's try to fit it. And when we try to fit it, we find that it fits the data very well. This very universal trend that we saw there, it fits very well with no fitting parameters, just knowledge that I have from what I put in the solution to begin with. And but this gives us some intuition, some physical intuition, right? I mean, because now when we are looking into this diagram, we understand that this is more like a, you know, like a pneumatic barrier, right? That allows us to compress and decompress the structure in solution. So it's more like a pneumatic barrier than a structured one. Something that will be very rigid, like a structured protein. And when you try to compress it, like with your arms, and you will try to compress it, it will break up irreversibly. And you want to have something that will compress and decompress as you put pressure on it. And so it's very important to distinguish between those two conditions. However, we have another problem. So we saw this diversity in low pressure. So we said to ourselves, okay, this is just disordered tails. So they are polymers, right? Like we were taught about in high school or something. So if I'll do a minor modification to them, a small mutation, the mean field should still work. Right? I mean, a small perturbation should not change so much the structure. So let's do a small mutation. We'll just cut out, we'll do a haircut for these tails. We'll cut out 5 amino acids. 5 amino acids is 1 nanometer out of a very long polymer. What will it do? So we cut 5 or 11 amino acids. So this is a charge diagram of the amino acid in this disordered domain. And we just cut out, you know, some amino acid in the end. And when we cut out the 5 amino acid, the black curve goes to the red curve. So we took a brush, we did a haircut, and the filaments extended. Right? I mean, usually when you would do a haircut, you would assume that things will collapse more. But here we did a haircut and something expanded. So this is ridiculous, right? This is just the opposite of what we thought would happen. Well, we didn't thought that would happen, but it was still very cool to see. Yes? Is the solid lines the theory or? Solid lines are theory that I will explain to you in a second.
So the theory you showed in the previous slide?
No, it will not fit. The theory before was just in the high pressure. So the theory for low pressure will need to be mended. Yes?
Is this for a given single protein?
Yes.
Or when you do other proteins, will you get the same behavior?
Probably not. Probably not. So we did it for a given protein for a particular reason that I will explain to you. Okay, so why is that? And why is that is because we, for many years, thought to ourselves that we have these proteins, and the reason that they are forming this gel is because they are handshaking, because they are making these bridges between them. This is why this gel is maintained. This is why you see them in a condensed phase. You see them that they are like holding hands one to another. And those cross bridges tether long filaments from one end to another. And that we know from statistical mechanics, we know how to solve such a problem, because this tail that is connected over here, we can just draw it with one tether over here. And now it's a problems of entropy, right? Because now when you are trying to push them aside, there is something that is holding it, because we are losing conformation, right? Then when a string is being stretched, it's becoming with less conformation that you can occupy. So it's an entropical problem, and we can write the entropy. But now the question is, well, where do we tether this thing? And this is depending on the different sequence where you can tether one thing to another. So this is the biology part in it, right? So we have one parameter here. Where did we tether the two tails from one end to another? And so we have one parameter. So let's try to feed this one parameter. And you can see here this one parameter, which I call biology. All the rest are physics. So I now have the high pressure and the low pressure regime. One that comes from mean field. One comes from, again, like polymer physics with a fitting parameter. And I can fit the entire thing. And I can do it for many, many other conditions as well. And it fits it with giving me one parameter, so to the strengths or to the locations of the connection between them. So how much time do I have?
We have 17 minutes.
17 minutes is good. Okay. So and here is biology. You asked me how much biology is making a difference. So we go into 500 million years of evolution. So yes.
So this K, you say it's the position along the strand where they
the position and the multiplicity of how many.
Of L or...
Yeah. So if you go into the evolution tree, you find out that from us to cows, there's not a big difference in the charge patterning of this neurofilament flow, but you can go down to zebrafish even, which is the positions of those amino acid charges are changing dramatically along the middle part of this disordered domain. But you can see that here in each one of them, at the end of the protein, there is a bunches of positively charged residues. And those positively charged residues is being conserved over 500 million years. Obviously, this is the figure that we put in the supplementary and we found out after we finished up the research. Okay, but this one is conserved. So something is conserved to make a sticker that can attack and make this tethering between those two filaments. So what did we learn? So we learned that biology or evolution, how to fine-tune these disordered domains to make the sequence to be functional. Functionality in a different way from lock and key. I demonstrated for you that disordered protein doesn't mean that you don't have disordered structure. You can make ordered structures with disordered domains. And I somehow hinted now that weak interactions between those proteins are very central to the functionality of those proteins. So I want to try to use it. And this is something that we just did recently. But I need to make a very short detour. And this short detour is to tell you that in our cells, we have another very important mechanism, and this calls the membrane, the engulfing walls that separated between something inside and something outside. It also have a lot of organelles inside our body, like the nucleus, that is protected by this membrane. What this membrane is made of? It's made out of usually lipids or other amphiphilic molecules. Amphiphilic molecules is just a molecule that has one part that likes water and one part that doesn't like water. And this is why they self-assemble to form all kinds of different structures depending on the condition. So again, through this hydrophobic hydrophilic hybrid, you can make membranes like you see over here. The hydrophobic parts, the oily parts, attract to each other, and the water-loving head groups are just facing the water in the surrounding. And you can think to yourself that according to the conditions that you will find, you will be able to make all kinds of different structures self-assembled in solution. So one of the key discoveries by Jacob Israelishvili, Mitchell, and Needham back in 1976 was that actually the geometry, again, here dictates a lot of the self-assembled structure, meaning the size of the head group versus the length of the tails, the hydrophobic tails, can dictate, in a sense, what are the structures that you can form. And you can understand it quite easily, right? If you have a very fat tail, a very fat head, and a very small tail, you make it into like some kind of like an ice cream cone. And when you try to assemble that in solution, you want the hydrophobic parts to be away from the water, so they will form some kind of a micellar structure. And when something is in the same, about the same, occupies the same volume, it will make something more lamellar, something more straight. So this is just coming from geometry. And this geometry really is important in day-to-day technologies, including the vaccine that you probably, I hope that all of you had in the last half a year or so. So in the Pfizer COVID-19 vaccine, we had an engulfing membrane that got RNA inside of it, and through the geometry of the lipids that were mixed up together, it made into these nanoparticles that were used to deliver the genetic code that made proteins in our body that were later on recognized and made our body vaccinated against COVID. Okay, so it's a very important, and very relevant to our day-to-day life today to understand how lipids or self-assemble to different structures. How do we look into structures? So there are many different techniques, including cryo-TEM, for example, or FRET that people are using. My lab is more specialized about this technique. It's called small angle X-ray scattering. Also, the result that you saw before was done with this kind of a technique. The technique is rather simple. You shine X-ray light and you look into the structures that are being projected, the Fourier transform that is projected on a detector very far away. What can you do with that? You can resolve the structure of the membranes themselves. Where are the head groups? Where are the tails? What are the dimensions that you have there? And you can also resolve mesophases, mesoscopic phases that they self-assemble into spheres, lamellars, hexagonal, tubes, whatever. You can understand the structure from the correlation spectroscopy that you have through X-ray scattering. Okay, so this is things that these are membranes, other membranes that we will measure before in different projects, obviously, but it allows us to differentiate between different structures and to understand the basic units, what is its structure through X-ray. So I won't go into much of the details, but you need to take my word for it. We can also use X-ray for many other parameters, if you want to say, and they are extremely used, this small angle X-ray scattering, to study intrinsically disordered proteins. So you can understand the radius of gyration, you can understand the molecular weight, what is the interactions between them. So there's a lot of information hidden in the X-ray scattering patterns. But again, from the lack of time, I won't get into that now. So this is the end of the detour. And now I want to do something. I told you I have intrinsically disordered proteins. I love membranes. So the next thing in line was to actually merge the two things together. And for that, I partnered up with very talented students in my lab, Guy and Tamara, and my lab manager, Gil. But we had to find very good friends and colleagues that came with additional techniques. Roy Amir is a chemist, an organic chemist, and he helped us with the chemistry here. Misha with the theory, and Ganit and Nathan with the cryo-TEM imaging. So we merge the two together and we term these two things together. We call it intrinsically disordered peptide amplifier. So basically we took a intrinsically disordered domain or peptide, and we glue it through chemistry. I mean, this is magic to me. And this is only, chemists know how to do it, to hydrophobic domains, to dendrimers. So you will see the terms 4 times 7 and 2 times 12. It's just what is the architectures of those hydrophobic domains. And we take those, and the reason that I told you before about this neurofilament is because the domain here, the disorder domain that we have over here, is actually inspired from this neurofilament law that I showed you before. So it will not form any secondary structure, but it will be a polymer. It will be a very charged polymer, and we want to see what happens. Now, the reason that we took this sequence of amino acid is because they are very charged. And you see that there is like a positive charge end groups and a lot of negative charge residues over there. The letters are just the one letter descriptions of what is the amino acid that you have over here. And what you can see here immediately that this is a charged proteins, but in solution, when you change the pH, you change the level of charges, the protonations of those amino acids. And specifically, you can see that around pH 6, something dramatic is happening, and the number of charged residues is dramatically reduced, and the level of charges in those peptides is changing. And this is how we will envision those self-assembled molecules, two polymers tethered one to another. One is charged, one is neutral. The hydrophobic one is neutral, obviously. The one that is charged is a peptide. So is it disordered? Yes, it is disordered. We can use other techniques, spectroscopic technique like circular diagonism spectroscopy, to find out that it's actually disordered. It's disordered in solution. It's disordered as a peptide. It's disordered in different pHs. And we know that. Okay, so we can measure to see that it's doesn't make any secondary structure. So disordered in all pHs. However, and this is the big surprise, what happens when you start to image them? And obviously we did the imaging later, but nevertheless, it's nice to see it in person. What happens when you change pH? When you change pH, you take for the different types of architectures, you find out that there's something very interesting coming. It starts from very circular and very well-defined spheres. And as you change pH, it's becoming to elongated tubes. And if you go even further, those tubes starts to aggregate. Okay, so they do a self-assembly to something that is well-ordered. Each one of those spheres is exactly the same as nearby it, although it's made out of intrinsically disordered building blocks. So it's a polymer, okay? It's jumping and dancing all around, but when it's aggregating, these jiggling polymers make it into a very well-defined structures. So we can see it with X-ray. So this is a scattering X-ray of those spheres, for example, has a very, very sharp feature. So usually when we work with polymers, and I work a lot with polymers, we don't get these kind of features. We get those features when we are working with solid nanoparticles. We get such a beautiful features. We can even see the turbidity of the samples. I mean, if you look at under bright light, you can see that when you change pH, it's at some point it's becoming turbid. So it started to make very long, elongated structures. And obviously we can see these elongations and making these tubes and aggregations, as we saw in the images, also through the X-ray scattering. And the X-ray scattering gives us something else. It gives us the mesophase that it happens. And here you can see something. We had a disordered domain, the same disordered domain, but now we changed just the pH, a different architectures of the tails. 4 times 7, 2 times 12. You remember I told you different architectures of how we made the hydrophobic domains. We get different condensed phase structure. So they form either hexagonal or lamellar structures that we can resolve from the X-ray. We can do something else. We can do an engineering, right? I mean, we have a disordered domain, something that is fluctuating between all kinds of things, but we have an amino acid sequence. And I told you, one of the beautiful things about amino acid is that you have a lot to choose from. So let's change the positions of 2 amino acids. We didn't change the total net charge or what kind of building blocks that we are using. We just switch 2 positions. And when we choose to what we wanted to begin with, to move this R with E just to replace them, we see that the mesoscopic phases completely change. We can do replacement, mutation, to take one of the histidine and replace it with glycine. Histidine is a charged amino acid, but it's becoming charged at around 6. When we do that, we fine tune when it's changing between cylinders to spheres by half a pH. So you can actually do a real engineering work to fine tune the interactions, and by fine tuning the interaction, fine tune the structure, the self-assembled structure. I mean, you throw it into water, it does it for itself. You don't need to invest any work into that. You don't need to put any energy into that. It does it by itself. We can do even further. We can take the same amino acid, same building blocks, and now segregate the positive amino acids to one end and the negative amino acid to the other end. Again, we're still using the same amino acid. This is how the scattering looks like. So this is for the expert. Moshe that is out there, Deutsch is in the audience on Zoom, can appreciate how beautiful it is. Moshe, right?
Absolutely right.
Okay, thank you, Moshe, from the audience. This is unbelievably beautiful scattering data. Why? Because it gives us a resolution that usually we see only with solids, to see such a perfect Bessel function like. And here are the fits and the data are colored. And what it tells us that the organization, because the segregation of the charges, make it into something much, much more dense, become very, very close packed because the negative charges over here are just attracting the positive charge residues on the other end, making like a helping. And that assemble into something very different. We can change the length of the amino acid. We can change the length, of the hydrocarbon chains, the hydrophobic domain. And when we change the hydrophobic domain, the transition between spheres to cylinders can be tuned dramatically from something that is starting at 6 and ending up at 5.9 to something that starts at 8 and ends at about 4.5. So you have a lot of tunability in the architecture, in a sense, of your building blocks to the self-assembled structure that you have in the end. And obviously you can do something with that. What are we thinking about? We're thinking about COVID today, every one of us. So we are thinking about how we can harness this self-assembly for our use. So one of the biggest problems with all drug delivery vehicles that we have in our industry today is that you have a double sword problem. On one hand, you want to make a vehicle that will carry the drug, will carry the RNA, will carry whatever, and it will wait until it goes to the target, will go to the cell that you want to infect with this drug or whatever, and not to spill anything on the way in your bloodstream. However, if you design something that is packing so well your drug or your RNA or whatever that you designed, when it comes to the cells, guess what? It doesn't spill it. And then you need to overload your system with a lot of copies and hoping for statistics to play its role for some fluctuations, and then it will do its work. But the problem is that when you overload things, you have a lot of toxicity because you introduce something that is not at the right amount that you designed to at the beginning. So here we wanted to use something else. We wanted to use the fact that when something is being engulfed into the cells, it tries to disassemble it. And the way to disassemble it is by changing pH. It makes it into very acidic environment. And here, when you make something very acidic, the structure is changing. So when structures are changing from a sphere to something very elongated, guess what? Things that inside are being spilled. So here we can use actually the mechanism that the body is having, the cells have to destroy it, actually to release as a release mechanism. And here exactly you see that. You see that when you do with a regular lipid-like structures or polymer-like structure. When you change pH, nothing changes. The spill rate is remaining. However, when you use our technology, when you change pH dramatically, you release much more.
But how can you control in vivo, I mean, this pH?
So in vivo, when you are engulfing the structure inside, it goes into a natural mechanism to make the environment acidic in order to disassemble everything that was inside. We can talk about it later. So this is just the take-home message. I know that the time is over. So we're using disordered peptides to make structures. We were extremely surprised to find that disordered peptide does not mean that we have heterogeneity in terms of the structure, On the opposite, we got really well-ordered structures. Those structures can be fine-tuned through understanding the interactions and how geometry is contributing to the self-assembly. And the structural phase transition might be even be useful for application as a release mechanism. And by that, I just want to thank those that actually made all of the work. Obviously, it wasn't me, but a very line of graduate students that helped throughout the years, and you for listening. Thank you.
Any questions? Yeah.
So it's so how will the immune system respond to such like myself?
So the immune system knows how to deal with that, right? I mean, obviously, our immune system got recognized, got us the small particles that we had in our COVID vaccine to be delivered. This is not the first time that actually we are using I'm not making any words, okay? How to design those to be stealth inside the blood vessels. And in fact, here we are introducing another functionality or another tool. So the amino acid themselves can be either used as a shielding to prevent attractions and recognition. So if you wanted to recognize a specific enzyme or specific protein, it can be designed for that. We have another project that we actually designed it with a specific sequence that can be recognized by a specific enzyme that cut it and then make the structural case. So this can be engineered in a sense to whatever need you need.
Yeah.
Was there any special mechanism to choose 1,000 out of 10 to the power 500 proteins? Because Earth's age doesn't look sufficient for statistical biology.
No, that's definitely. So that was related to another. So how can you choose? So the way that I mean, there are different strategies. People are either using something very simple. Let's take 2 amino acids and play with them. Okay?
No. I'm asking about how did the biological systems choose the 1,000 proteins out of possible 10 to the 500?
Oh, how does biology do that? Evolution. Those things that I mean, I guess it was a very long game of throwing dices, and only those that had a bigger strike of and won and survived. And so biology and evolution are just trying a lot of different combinations, and those that fits, those that makes it into something functional, is being remained in our environment because they can be used. So this is our understanding of how evolution is being progressed throughout the years.
But it's a bit confusing because the Earth's age or the age of evolution doesn't look sufficient for that statistics.
It's not very efficient, and that's why it took millions of years. It wasn't something that was done on a 7. Okay, maybe not 7 days, but maybe yes. I don't know. So it took a lot of time, but
we don't really know. I mean, nobody knows.
Nobody really knows the answer.
Whether it makes sense or it doesn't make sense.
It is what it is. Yeah, it is what it is. It's not efficient. Okay, it was not planned to be efficient because it's a game of luck. Okay, but in this game of luck, it made it into us eventually.
So I mean, this business of self-assembly of peptides I mean, this is already a well-known subject. So I think that the new part in your case was that you actually use electrostatics, manipulation of electrostatics, right?
Yeah.
Because otherwise people normally think about more about hydrogen bonding and stuff like that.
Yeah.
Okay, that's true.
Yeah, so electrostatics here plays a major role in the things that I was working with, a very small subset, obviously. The one thing that we do know, and we have a debate on it, but it's a healthy debate, is that many of the intrinsically disordered proteins are charged. And the reason is very simple. They are charged because charged polymers and hydrophilic polymers do not tend to collapse on themselves. And if you want to have something that is fluctuating and do not aggregate, you want something that will be able to get away from the van der Waals attraction. And for that, you need to have something that will do the repulsion. And the repulsion is nothing else is there in the chemistry of polymers than electrostatics. So that's the only thing that prevents it from collapsing and make it into like a global. And that's why I think IDPs are notorious to being charged and hydrophilic.
Right, but you know, this argument of repulsion basically says that you would have just one sign of charge. So
that's correct. So it's but this is on the first order. On the second order, it's becoming complicated. Electrostatics with the polyampholytic nature is making it into something interesting too.
Okay, so with this we're out of time, and let's
All
thank
right.
the boy again. Thank you very much.